Good afternoon. Thank you for coming to this session on angiogenesis. I'm really excited. We have 4 speakers today who are going to give quite a diverse and forward-thinking perspective in terms of the field of exercise and angiogenesis. The first speaker today is Dr. Katrien Bock from ETH Zurich, and she's going to speak on endothelial metabolism in the muscle.
So I would like to start off by thanking the organizers for inviting me here today. Very happy that we are able to fill a complete session on muscle angiogenesis, so I think we have a field now, actually. So for me, it's the 3rd time that I'm attending an IBEC meeting. Back in 23 when I was in Maastricht, being trained as an exercise physiologist, and then went to Seoul to somewhat present my PhD work there. And then I was away for a very, very long time, and my brain was completely reconverted into vascular biology. So my lab, what my lab is now doing is I basically combine what I have been doing during my PhD with my postdoc and put it together in a lab where we really focus on looking at the vasculature or endothelial cells in the muscle. And the reason why I'm so interested in looking at vessels in the muscle, there are 2 main reasons for this. One reason is that the vasculature, and we have seen this picture before earlier in the morning, is the vasculature really provides a niche where other cells like to reside. You see endothelial cells here, and then you have macrophages and fibroblasts and pericytes, and all of these cells reside in the close vicinity of endothelial cells. And we really think there are interactions between those cell types that are extremely important for the maintenance of muscle homeostasis. And actually, the second part of my talk will be on this vascular niche. The second reason why I'm so interested in vessels in the muscle is because exercise is one of the very few physiological means by which one can stimulate angiogenesis in an adult setting. And this is really important because at this moment in the field of vascular biology, it is somewhat the holy grail to find means to promote revascularization during settings of regeneration upon injury in elderly or organ transplantation. So if we could understand how exercise promotes angiogenesis in the muscle, well, maybe we can then find means to also promote angiogenesis under a regenerative setting. And it was actually with this concept in the back of our heads that we really embarked on a big study where we tried to identify or try to study how blood vessels grow in the muscle. And part of this work is published and was led by Zheng Fang in my lab and is now taken over by Guillermo Turiel. So what we did is we did single-cell sequencing assays of isolated CD31 positive, CD45 negative endothelial cells under sedentary and after exercise conditions. So those are the cell populations, or those are the cells that we came up with, and you can see that there is nice overlap between sedentary and exercise conditions, and that you have some additional populations popping up here upon exercise, which are the cells that proliferate and migrate and are responsible for de novo vessel formation. And if you then really do an automatic annotation of these, or an automatic clustering of these endothelial populations, what you actually see is that they nicely and automatically align alongside an anatomical axis as is indicated here. So we found arteriolar endothelial cells, we found venous endothelial cells, and we also found several capillary endothelial cells, cell population. One of those is very peculiar. We don't really know what it is. It has capillary markers, but it's completely separated from all other endothelial populations. But we really got interested in those endothelial populations here. And what we found is when we really looked at these different endothelial or capillary endothelial populations, and is that those were characterized by differential expression of the activating transcription factor 4 and its downstream target ATF3. And those are transcription factors which are known to be involved in stress responses in many cell types. And this was really, this became really interesting when Guillermo also did the pseudo-time analysis, and he really showed, or this pseudo-time analysis somehow suggested that whenever endothelial cells become angiogenic, that those endothelial cells are derived from the ATF3/4 positive population. So this capillary population here, this one, this has high ATF3/4 expression, and the one a bit separated from this AV axis is very low in ATF3 and ATF4 expression. So we really then wanted to get some better insight into an anatomical localization of these different capillary endothelial cell populations, and we decided to stain for ATF3 on muscle sections, and we did this in oxidative muscle area. So what we mostly do is we take a gastroc and then we really localize in the oxidative area and the glycolytic areas. You can see it here nicely by fiber type staining, and then in red you have the capillaries, and here in white you see the staining for ATF3, and you can nicely see that in the oxidative area there are more ATF3 positive capillary endothelial or capillaries when compared to in the glycolytic area, where we found only very few double positive cells. And you can actually see it much better when I show you some longitudinal sections where you see the oxidative fibers here, and you can nicely see that there are plenty of ATF3 positive endothelial cells in the oxidative area, whereas there are very few ATF3 positive cells in the glycolytic area, and if they are ATF3 positive, they are mostly non-endothelial cells. So we actually think that these ATF3 positive endothelial cells are red muscle endothelial cells or oxidative muscle endothelial cells, but for the rest of my talk I will call them red muscle endothelial cells. And we also confirm this with more, let's say, crude assays where we just isolated the gastrocnemius muscle and then physically separated the oxidative area from the glycolytic area, isolated the endothelial cells, brought them in culture, and then subsequently stained for ATF3. And you can see here the red muscle endothelial cells are highly ATF3 positive, whereas the white muscle endothelial cells, only very few ATF3 positive endothelial cells were found. So ATF3 positive endothelial cells are red muscle endothelial cells. Now, to get a better insight into the transcriptional differences between those two endothelial or capillary subpopulations, we subsequently repeated this assay. So we isolated endothelial cells from red versus white muscle, and then subjected them to RNA sequencing. And here on the left side, you can nicely see the top differentially expressed genes, and ATF3 and ATF4 nicely pop up in this list of differentially expressed genes. But if we then subsequently did the transcription factor motif enrichment analysis, what we then found is that ATF4 was the most differentially enriched transcription factor. And this somehow suggested that besides marking different endothelial populations, that ATF4 might also have a functional role in those red muscle endothelial cells. Now, what do we know about ATF4? Well, ATF4 is a transcription factor that has been heavily studied in a context of cellular stress, where it acts downstream of EIF to alpha. And subsequently activates an adaptive response inside the cell. But recent work from the Manning group in Harvard, they also showed that ATF4 under some specific condition is acting downstream of mTORC and crucially contributes to the anabolic response inside cells through the activation of a specific set of anabolic genes, mostly involved in the uptake of amino acids, the synthesis of non-essential amino acids, etc., etc. Now, it's known that ATF4 acts dimerizes and has specific or binds to other transcription factors. So the genes to which it binds, those are really dependent on the specific conditions and on the transcription factor it dimerizes to. So also, the Manning group, what they subsequently did is they identified an anabolic gene set, so those genes which are activated by ATF4 under conditions where it's downstream of mTORC, and they also identified and described this adaptive gene set, so this stress-responsive gene set, which is activated by ATF4 and its unknown co-transcription factors under conditions of stress. And if we use these gene sets and then map them to our own gene set, what we found is that there was a huge difference between red and white muscle endothelial cells in the anabolic gene set, where white muscle endothelial cells had much lower expression of anabolic genes, whereas we didn't really find any difference in the adaptive gene set between our two endothelial cell populations. So ATF3 for capillary endothelial cells are red muscle endothelial cells. Red muscle and white muscle endothelial cells show transcriptional heterogeneity that might be controlled by ATF4-dependent anabolic signaling. So we subsequently wondered whether red and white muscle endothelial cells are functionally different, and if yes, if this could be determined by different ATF3 or ATF4 signaling. So to do so, we went back and isolated endothelial cells, brought them in culture, and then did spheroid sprouting assays. And what we do then is we make a small ball of these endothelial cells, put them in a hydrogel, stimulate them with VEGF, so the main angiogenic growth factor, and then we can really analyze the sprouting of these endothelial cells. And you can already nicely see in the images here that white muscle endothelial cells form very few sprouts and very short sprouts, whereas the red muscle endothelial cells have form much more sprouts and much longer sprouts. So to then really dissect the functional role of ATF3 and 4 in red versus white muscle endothelial cells, we used, I forgot to say that this was dependent on proliferation, but it's not that important. So to dissect the functional role of ATF3/4 in this phenotype, what we subsequently did is we used HUVECs, and we knocked down ATF3 and 4 in HUVECs via lentiviral delivery, and we basically repeated the same assay. So you see the sprouting assay here, and you can nicely see here that upon ATF3/4 knockdown, the sprouts are shorter and we have less sprouts. And also here, this was dependent on proliferation, because if we measured the number of proliferating cells, there were much more proliferating cells in the control conditions versus the ATF3 for knockdown conditions. Now, finally, to test the functional relevance of ATF4 for our observations, what we did is we used a doxycycline-inducible ATF4 overexpression construct, which really allowed us to carefully titrate ATF4 expression by increasing its levels by twofold. And if we did this, what we saw is that this did not affect the proliferation rate of red muscle endothelial cells, which already proliferated quite significantly, but it increased the proliferation rate of white muscle endothelial cells. So this means that basal ATF3/4 primed endothelial cells for angiogenesis. Now, since we since we already saw in our RNA sequencing data that there were big differences in anabolic genes between red and white muscle endothelial cells, we subsequently wondered whether the differences or the angiogenic differences between those endothelial cells were dependent on metabolic rewiring in endothelial cells. So we first checked for the expression of all amino acid transporters, and you can see here that they were downregulated upon ATF3/4 knockdown, and this effectively led to a decrease in amino acid uptake in endothelial cells. And we also saw lower expression of all genes which are involved in the synthesis of non-essential amino acids, such as serine, such as glycine in endothelial cells. And also here, we confirmed by 13C glucose tracing that there was a lower incorporation of glucose into serine and glycogen, actually showing also on a functional level that there is a broad rewiring of amino acid metabolism in ATF3/4 knockdown endothelial cells. So reduced import combined with reduced de novo synthesis of amino acids somehow suggested that those endothelial cells had a lower ability to maintain intracellular amino acid levels, and this was indeed the case, because if we just measured amino acid levels inside the cells, we saw that almost all amino acids except for serine, which apparently endothelial cells very efficiently take up from the medium, that all of those were reduced upon knockdown of ATF3/4, and this resulted in lower protein synthesis in those endothelial cells. So finally, if we have lower endothelial cells, lower intracellular amino acid levels, if we then increase the levels of amino acids outside of the cells and literally push amino acids to enter into the cells, can we then somehow rescue this phenotype? And this is what we did, and this was indeed the case, because if we supplemented our normal cell culture medium, which already contains high amino acid levels, if we supplemented with additional non-essential amino acid or essential amino acid mixtures, what we saw is that non-essential but not essential amino acids could completely rescue the proliferation phenotype in our cells. So ATF3/4 metabolically prime red muscle endothelial cells for angiogenesis. And we finally asked whether ATF4 is then required for exercise-induced angiogenesis in vivo. The reason why we only why we suffice to knock down ATF4 in vivo and need ATF3/4 knockdown in vitro is because for some unknown reason, those two are redundant in vitro in endothelial cells, so you need to knock down both of them to see a knockdown of genes, whereas ATF4 knockdown in vivo suffices to see a complete blunting of anabolic genes. So this is what we did. We knocked down ATF4 in vivo using a PDGFB CRIA32, so a vascular-specific inducible driver, and you can see here that is completely wiped out ATF4 expression in endothelial cells. We isolated again endothelial cells, did RNA-seq, and you see the PCA plot here. So those are the red muscle endothelial cells versus white muscle endothelial cells. And if you knock down or knock out ATF4 in these cells, you could actually see that red muscle endothelial cells become completely indistinguishable from white muscle endothelial cells, showing that ATF4 is required to maintain red muscle endothelial cell identity. And this is also what is shown here. So if we look at the anabolic gene set or ATF4 dependent gene set, you can see that there is not a single differentially expressed gene anymore when we remove ATF4 from endothelial cells. Now, finally, we still needed to do one more experiment, and what we did is to look whether ATF4 is required for exercise-induced angiogenesis in vivo. And what we then did is we used those mice, put them on in a running wheel, allowed them to run for 2 weeks, and looked at exercise-induced angiogenesis. And this is what you see here. So you can nicely see an increase in vascular density in a normal wild-type mouse, and this is completely blunted upon loss of ATF4 in endothelial cells. And this is the data of endothelial cell proliferation. So in conclusion, ATF3-4 muscle endothelial cells represent a subpopulation of endothelial cells which are metabolically prepared for angiogenesis. There were a lot more data in the paper, but due to interest of time, if you want to know more, please check it out. It's published meanwhile. So I quickly want to touch upon another project that we have been working on recently, completely unpublished and actually a bit of a puzzling project at this moment. And this project basically started off quite some time ago when I attended the Bengt Saltine Memorial Symposium, and I saw, I think it was Erik himself, presenting some data on a review that he had been writing together with Adam Rose, where they discussed intensively about how muscle takes up glucose and how all of this is controlled by GLUT4 and GLUT4 translocation in muscle. And in this cartoon, he somewhat showed, and actually based on all data from the Pappenheimer somewhere in the 50s, where they suggested that glucose just passes the endothelial barrier and enters the muscle interstitium. And this, for me, was very, very difficult to believe, so I went back to Belgium at that time and decided to generate an endothelial knockout, GLUT1 knockout, which is the main glucose transporter in endothelial cells, to study whether glucose is or is not transported over the muscle endothelium, and whether endothelial GLUT1 contributes to muscle glucose transport. Now, before I continue with this, showing the data, I have to say that all of this work is done, or most of the work in my lab is done by Jing Jiang, a talented PhD student, but in fact also Erik could stand here and present it, because this is a very intense and has been a very intense and full collaboration. So what we did is we made a GLUT1 endothelial knockout, and then Erik clamped the mice to really check or evaluate whether endothelial GLUT1 is required for glucose transport over the endothelium into the muscle. And this is glucose infusion rate, and you can very rapidly or immediately see that loss of GLUT1 in endothelial cells reduces glucose infusion rate into the muscle, and this is the quantification of those data. So we were very happy by this, because we found reduced insulin-stimulated glucose uptake in muscle, gastrocnemius, and soleus, but actually we didn't see any effect in other metabolic tissues such as white adipose tissue. So we immediately wanted to finish this project, and Erik and actually Kim spent a lot of time setting up means to really sample interstitial glucose levels to finally show that there was a difference in plasma versus interstitial glucose levels, because if GLUT1 would contribute to glucose transport over the endothelium, one would expect that if you remove GLUT1 from the endothelium, there would be a drop in interstitial glucose levels. Unfortunately, this was not the case. So these are the data from those experiments. So GLUT1 is not required for glucose transport over the endothelium. This was really puzzling, so we wondered whether loss of endothelial GLUT1 would induce muscle insulin resistance, and this was indeed the case. So this is one quantification of those experiments, but we checked several kinases involved in glucose uptake, or insulin signaling and glucose uptake in the muscle, and you can nicely see that GLUT1 deficient endothelial muscle becomes insulin resistant. So what is the mechanism for these observations? Well, when we were checking or thinking about this, we obviously immediately remembered our own work, which we published a few years ago, where we observed in the brain during development that if you knock out GLUT1, that there is microgliosis, astrogliosis, so a pro-inflammatory phenotype. And obviously, we were aware of the work of Mark Fabbraio and Bente Pedersen, showing that also inflammation in muscle can contribute to muscle insulin resistance. So we analyzed immune cell populations in the muscle. What we found is that upon loss of endothelial GLUT1, there is an accumulation of macrophages in the muscle in the absence of an increase of monocytes. And this is very interesting, or this I found very interesting, because in our other experiments, usually when we have muscle damage or ischemia, and monocytes are recruited into the muscle, we always see an increase in monocytes. And this actually suggested that what we saw was a local increase and a local proliferation of macrophages in muscle. And this was indeed the case. So we saw increased proliferation of macrophages in the muscle. And then we wanted to evaluate whether this were recruited macrophages or whether those were resident macrophages from the muscle. So we deleted all monocytes from the blood, so preventing monocyte recruitment into the muscle. And what we found is that despite the lack of circulating monocytes, that the increase in macrophages was still there, showing that the phenotype we observed was due to the activation and proliferation of resident muscle macrophages. Now, we did some further phenotyping on these macrophages, and we actually saw that this was mainly caused by an increase in the number of LIFE-1 high macrophages, also a specific macrophage population in the muscle. And when we stained these macrophages for these macrophages, you can nicely see here that those are, really embracing the vasculature, perivascularized. So these guys here all in magenta, so they are perivascular macrophages. There is no description on what they do in the muscle. It suggested that they contribute to muscle homeostasis, but there is no real evidence for this. Now, to finally show whether macrophages really are responsible for the impairment in insulin sensitivity, what we finally did is we used an anti-CSF1R antibody, and an anti-CSF1R leads to anti-CSF1R antibody treatment leads to the depletion of macrophages in muscle and in more or less any other tissue. So this is what you see here. So when we treated those mice with this antibody, we could see that insulin sensitivity was completely rescued, backed up by the insulin signaling data. Two final questions. How does this work? Well, we know that this is dependent on endothelial crosstalk, because if we isolate endothelial cells from these mice, add the conditioned medium to macrophages, we see that those start to proliferate. So this is really angiocrine crosstalk. The endothelium directly controls the identity and function of resident macrophages in the muscle. And then second, if we see that these resonant macrophages contribute to the development of insulin resistance in the muscle, do we then also see activation of resident macrophages upon high-fat diet? Well, this is indeed the case, because if we feed mice a high-fat diet, so there is a bit of a fuss in this slide here, what we see is that GLUT1 mRNA goes down in muscle endothelial cells. We see an increase in resident macrophages in the muscle, and these macrophages are locally proliferating. So, conclusion number 2, final slide. First, we still don't know whether the endothelial or the endothelium actively controls muscle glucose transport. Second, the role of the vascular niche in muscle homeostasis and insulin sensitivity is likely more important than initially assumed. And third, altering the niche function through affecting endothelial metabolism, either through loss of GLUT1 or upon a high-fat diet, leads to the activation and proliferation of resident macrophages and insulin resistance. Thanks for your attention.
Thank you very much, Katrien, for that interesting talk and some new insights into endothelial cell behavior. Ola, do you have a question?
Yeah, thank you so much for the presentation. I have two questions. The first one is about the connection between ATF4 and mTORC1 and GCN2. Because the two of them, GCN2 and mTORC1, they are typically activated under different scenarios. So for example, you don't have amino acids, GCN2 is activated, you have amino acid, mTORC1. So can you reconcile why both of them, will activate ATF4?
It's a good question. I don't know, and I don't think this is something that is very well known in the field. In addition to this, most often the role of ATF4 is studied under in vitro conditions, which we already know is completely different than an in vivo setting. We are trying to now develop some models that would allow us to really see when ATF4 gets activated in muscle or in endothelial cells in vivo, what are its binding partners, which gene does it activate. So I hope to be able to address your questions in a few years.
Like, is ATF4, is it phosphorylated, like the interaction with GCN2 or mTORC1? Because
yeah, no, and so it's basically, it's alternatively described under stressful conditions. There is post-translational control, but I think it's phosphorylated, but I'm not so sure whether the phosphorylation sites are really important for its function or for the selection of its binding partners.
Okay, thank you. So the second one is about the GLUT1 deletion in EC, in endothelial cells and the impact on glucose infusion rate in your CLAMP study there. Did you actually measure GLUT4 expression in those, in the muscle of those? Because if I don't know, you said it's not published yet, but I think it's a question that you should probably think about in terms of whether it shouldn't be, but what impact does GLUT1 deletion have on GLUT4 in muscle?
So we didn't measure GLUT4, but the fact that our rescue experiment completely rescued glucose infusion rate somewhat argues against a difference in GLUT4. We did insulin receptor binding assays, those were not affected.
So in your study, you definitely demonstrated the importance of amino acid metabolism for endothelial cell migration and proliferation. Mm-hmm. But my question is that you didn't necessarily address where the sources of amino acids would come from. So my first question would be that, would you suppose that skeletal muscle would be releasing these amino acids in response to an exercise stimulus? And my second question would be that you demonstrated that in your model, the exercise stimulus obviously elicited an angiogenic response, but perhaps would you expect to see an increased or a greater angiogenic response if you induced maybe perhaps eccentric muscle contractions, inducing greater muscle damage and greater amino acid release?
First answer, I don't know. Second answer, I also don't know. Potentially. Good suggestion and experiment we need to do. But I don't have any clue about the source of the amino acids, whether they are muscle-derived or derived from the bloodstream. We don't have a clue.
Very nice talk. So you showed nicely that ATF4 is sufficient to drive proliferation of white endothelial cells, right? So does it also drive the heterogeneity that you see between white and red endothelial cells?
So we know that so if you knock out ATF4, there is no difference anymore between a red and a white muscle endothelium.
Yeah, I'm talking about the sufficiency experiment, so your gain of function experiment where you overexpress it. So is it sufficient to drive the heterogeneity that you see? Or is it does it just drive proliferation?
Yeah, so this is difficult. In our in vitro setting, we see differences in proliferation, right? In vivo, under quiescent settings, we don't see a difference in proliferation between ATF4 positive and negative endothelial cells because they simply don't proliferate. So it's difficult to answer whether this is really because of. I think you need a second activator. You need VEGF or you need the pro-angiogenic agents, and to really push angiogenesis, ATF4 just ensures that the metabolic machinery is there to take off fast.
And then one really quick question. So when you delete ATF4 in the endothelial cells and you show that there's a block of exercise-induced angiogenesis, do those animals run the same?
Yes. So we did two weeks of training. They perform the same. They have the same insulin sensitivity. They have the same increase in oxidative capacity, but we didn't go further than 2 weeks. We still need to do this experiment.
Great. One really fast question.
Okay, too fast.
Oh, okay.
These red and white endothelial cells, you observe them in rodents. Do we see them as well in human skeletal muscle?
Yeah, so the thing is, so we try to now assemble every single human data set we can find using mostly single nuclear sequencing, because mostly people do nuclear to look at muscle, and then very few endothelial cells are picked up. And we are really putting together the data set to see whether we get sufficient cells to really look at this. Our own data, but this is in PED and control patients, seem to suggest it is, but I don't want to put my hand in the fire for this at this moment.
Okay, thank you. One quick more question. If you get into a situation where you boost capillarization, for example, prolonged training, or you get the opposite, unloading in rodents or detraining, do you expect or do you know if you're going to change the proportion of these white and red endothelial cells?
We know that in exercise conditions, we see an increase in ATF4 positive endothelial cells because they are the ones that proliferate, right? Under unloading conditions, we have not been looking at those.
Okay,
It's
thank you.
good.
Okay, thank you very much, Katrien.